This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 22 for February 27th, 2009. From New York City, I'm Vincent Racaniello, and joining me today from our northern neighbor is Chris Upton. Welcome to TWiV, Chris.
Hi there. Good to be with you.
Northern neighbor, of course, being Canada. Chris is a professor at the University of Victoria in Victoria on Vancouver Island, Canada. What a great place to have a job, right?
That's right. It's pretty nice out here. We're in the sort of lotus land, as the Canadians like to put it.
I've been once to Vancouver Island. It's just beautiful. You're a lucky guy. How did you end up in Vancouver, on Vancouver Island?
Vancouver Island? I did my PhD at UCL in London. No, not actually. Sorry. I did my PhD in London and applied for a postdoc in Edmonton with Grant McFadden and went out there in 1982 and stayed around there for quite a while and then finally figured out I should get a real job, or as much as close to a real job, and applied for a faculty position out here and got lucky.
So you're content there?
Yeah, it's pretty nice.
It's great. Well, the reason we have you here today, I think you were one of the first listeners of TWiV, in fact, many months ago, and you came on my radar screen over Twitter, of course, and you're a virologist, and your interest is pox viruses, right?
That's right.
Because that's what you were doing with
Grant McFadden.
Grant McFadden, thank you, who's now in Florida, correct?
That's right.
And you're also a bioinformatician, a viral bioinformatician. So I thought we would talk a little bit about that as well.
Okay, sounds good.
So we have a couple of stories as usual. Oh, I wanted to ask you, so you gave your virology class an exam yesterday, is that correct?
That's right.
Did you get any wacko answers?
One or two. One of my questions is calculate the mass of a tobacco mosaic virus virion.
Uh-huh.
And so the idea is that they're allowed to sort of guess what the size of or mass of nucleotides are and stuff. And one guy in his estimate said, well, it's 2 Daltons. I thought, oh, no.
That's funny. A couple of hydrogen atoms, huh?
That's right. That's right. But he still managed to get an A-. I don't know.
It's always good to know your chemistry as a virologist, I can tell you.
That's right.
Anything else?
Not too much. Most of them did pretty good. It's a midterm, so there's some easy stuff on there. A few things that we use multiple choice in class with iClickers to sort of get instant feedback. So I use some of those same questions on the exam to make sure they've actually been listening to them, and they do pretty good on that.
Yeah, I like the clicker technology. I was at a meeting that used that, and I thought it was great. I'd love to use that in a course. So you have them with the clickers during the course, right, as you're lecturing? That's
right. So each lecture I have 2 or 3 questions, and I guess part of the philosophy of them is that it's not a perfect question, and that it's supposed to be a couple of possibilities in the answers, which makes it easier to write the questions. And so you get a spectrum of answers, and then one thing you can do is say, you guys are way off mark, talk to each other for a minute, and then they come back with another answer, and you see if they've convinced each other. It works well.
I'd love to use that when I teach an undergraduate course next year here at Columbia. I'll be in touch with you to find out how to get that to work.
All right, yeah, the technology is really easy as well. That's one thing we like here.
You need to buy all of this. That's the limitation.
Well, the students need to buy the clickers.
I see. And you have some kind of receiver in the classroom, correct?
That's right. But the iClicker company throws in a receiver for every 100 clickers that are in the course, sort of thing.
How much is a clicker?
In the US, I think it's about $30 or $40.
Wow. A small textbook.
That's right. I mean, if you're going to use it for 3 or 4 years, it's relatively small peanuts.
Well, maybe some of your students will listen to this podcast and hear this little discussion about their exam questions. Well, we hope you did well.
That's right. Well, I used the podcast as part of the course. There's a couple of times I said, go listen to this podcast and make a comment. It's part of our online work.
Mm-hmm. Yeah, you have a great online part of the course there. I've visited it a few times, and I really like that as well. I think you're using all the Web 2.0 tools to teach virology. That's great.
Right, we're trying.
Okay, let's do a few stories here, and then we'll talk about viral bioinformatics. There has been an outbreak of hepatitis B in India in the past few weeks, and I caught this in the news and also on ProMedMail. The latest ProMedMail bulletin is February 24th. 43 deaths. There are 43 deaths so far. It's the Sabarkantha district of India. They're undertaking a massive immunization campaign. There is, of course, a hepatitis B virus vaccine that's been available for quite some time, and they have one campaign in India where they're immunizing 60,000 people. The origin of this outbreak is a bit enigmatic. I have seen it blamed on contaminated needles used by physicians. Another article suggested there might be a mutant virus strain because it's particularly lethal, but I see no evidence, no scientific evidence for that. This virus is spread by blood and blood products. It can also be spread at birth, and it can be spread by sexual activity. So I don't know which of those is factoring in this outbreak.
That's right. I haven't really heard anything about it, and don't know an awful lot about Hep B. In fact, I only got a vaccination myself last year.
Yeah, it's for people working in hospital settings, it's pretty important to get one. I've never had one. My colleague was chastising me the other day for never having gotten one, but it turns out it's a 3 immunization course, correct?
Yes. I did it funny because I needed it in a rapid way. You can do it as sort of a rapid process, and that actually takes 4. You do 3 really quick ones, then you go back a year later.
I see.
For your final booster.
That is rather expensive, and it may be why it's not extensively used in India. Right. India is endemic for hepatitis B. There's about a 2 to 7% infection rate in the whole country, and that's about 40 million infected people in the whole country. Of the 25 million infants that are born each year in that country, over 1 million run the risk of developing chronic, that is, lifelong HBV infection, and 100,000 people in India die every year because of HBV. So this is a significant infection. In fact, I think globally 1 million people die a year of HBV. And its complications. So, I mean, the solution is the vaccine, but as I said, it's expensive.
That's right. I'm not sure why there hasn't been a product produced that links the antigen into a live virus. That might make it cheaper.
Right. Yeah, these are all virus-like particles that are parts of this vaccine, so they're rather expensive to make. I don't know the extent of routine immunization in India. It may be very low, but obviously they do have a vaccine because they're doing mass immunizations now to try and stop this. So we'll keep an eye on this and see if we can find the origin, because right now we don't really have any information. Hep B, of course, is an enveloped virus with a DNA genome and a very unusual life cycle. It goes through an RNA intermediate, and reverse transcriptase is part of the replication strategy. It's quite unusual. So that's hepatitis B in India. So we'll put some links in, but ProMedMail has some recent information. Our next story is about a gene therapy trial for HIV or AIDS. Have you seen this one, Chris?
Yeah, I just looked at the paper. It's kind of interesting. Some kind of big questions. I'm not sure how it fits in with the sort of drug use, not drug use, but drug therapeutics.
Right. Well, as everyone probably knows, HAART is a multi-drug treatment for AIDS, and quite effective, actually, for prolonging life. You use 3 or 4 drugs at a time, antiviral drugs. It's good, except, of course, you have to take your drugs every day, and they're toxic, and you do get emerging mutants. So if something better came along, we would adopt it. And this paper suggests the use of a ribozyme, which is an RNA enzyme. So ribozymes are found in introns and tetrahymena, bacteria. They're RNA sequences that can cleave themselves. So there's an enzyme without no protein component, an RNA component. And they can also be engineered to cleave other RNAs. So in this case, what they did is was previously done to design a ribozyme that would cleave the HIV-1 genome in a very conserved region. And previously it was shown that this ribozyme could knock down virus replication. The study that we're talking about, this is an amazing study actually, because what they do they had 70 some odd patients in this study, and they took out blood and purified peripheral stem cells, blood stem cells from these patients. And for each patient, they took those cells and introduced a retrovirus carrying the ribozyme into those cells, waited a week or so, and then put the cells back into each patient. It's not exactly a bone marrow transplant, but they're adding these new stem cells, which they hope will develop into lymphocytes, which are the targets of HIV. That's incredible. This is, I think, the biggest gene therapy trial of this kind ever done. You can imagine the logistics and so forth. But what they found was quite interesting, that these patients, they had two groups. One received the ribozyme and one a control virus, or I should say virus infect blood stem cells. And those who got the ribozyme had lower plasma virus levels over the course of the study, which was something like 100 weeks. Huge. I can't imagine doing this.
That's right. It's a huge amount of work.
It's a huge amount of work and money and patience and lots of physicians and nurses all involved in this, as you can imagine. So these patients who got the ribozyme had less virus, and they also, their CD4 cells expanded more than the control group. So that's good. It suggests that the ribozyme-containing stem cells differentiated into lymphocytes in these patients, and these became resistant to infection and propagated in these individuals. And what they say in this paper is that this has great potential for treating AIDS. I don't know practically how much potential it has. I don't have enough expertise to talk to that, but from the virological viewpoint, it looks pretty promising.
I thought it was interesting. I was a little confused in that there was very few cells, less than 1% in the blood, that were positive for the ribozyme, that they could detect it in. Maybe thinking about detection, maybe there's a lot more than that. But if there's only that many cells different or protected, you'd think the HIV would be able to replicate in the other 99%, and you wouldn't see much level change in the virus. I don't know. It's
well, the level change, admittedly, was very small. In fact, they say they say it was statistically really not significant, but consistently lower in the ribozyme treatment group.
But at first stage, I guess that's better than being higher, right?
Right, exactly. One thing we should point out is that they actually took these patients off heart for, I think, 4 weeks to get levels of virus back up again.
That's right. It's a bit of a hard sell.
Well, you have to agree to do that, of course, if you're entering this study. And, you have to be a very dedicated patient. So I applaud these patients who participated in this study. But the idea of that was to get the virus up, which would then infect all lymphocytes, and presumably it would select for enrichment of the ribozyme-expressing ones, which would not be killed by the virus. So that was pretty interesting. The last aspect of this that I found very curious is that they didn't observe any viral mutants in the patients that received the ribozyme. You would think that one base in the targeted sequence in the genome would be sufficient to make resistance to the ribozyme. HIV is an RNA virus and makes many, many mistakes in its replication. So the fact that they got no mutants is very interesting. It must be that they used a conserved region of the genome. So mutations produce a less fit virus, which doesn't survive selection in the patient. So that's good.
That's right. I'm not sure how big a target is for a ribozyme. So how much of a region of the virus would have to be conserved to still allow it to be cleaved?
Yeah, I don't know the target offhand, but I do know that one base is enough to get resistance to cleavage. And you would think that, this virus, of course, makes millions of mutants a day in a patient, and I'm sure there are among those ribozyme-resistant mutants, but apparently they're not fit.
Well, one reason might be that they were targeting TAT and VPR.
Right.
So if the ribozyme hits very early transcripts of HIV, so if you don't get TAT made, then you don't get virus made, then it may be able to block virus production at the very early stage, so you don't get a lot of variants. That may be the key.
Yeah, before a lot of replication can occur.
That's right.
Yep, that may be another component, sure. So this is pretty neat. We'll put a link to it. The paper was published in Nature Medicine just recently, I think last week or so, possibly the way it's going to go in the future unless we find ways to deliver a ribozyme without having to take out blood stem cells. The next non-pox virus story has to do with influenza. A paper came out a week or two ago on antibodies that apparently neutralize many different subtypes of influenza virus. As everyone knows, we have to get a vaccine every year against influenza virus because the virus undergoes antigenic variation. It's very structurally plastic, and it can accommodate changes that basically abrogate antibodies being able to neutralize the virus. So the goal is, of course, to somehow make an influenza vaccine for life, one immunization that will protect you forever. Did you get a flu vaccine this year, Chris?
I've missed it this year. The last probably 4 or 5 years I've got them. I've been convinced that I should, but I just missed the couple of days at the university. I never got around to it.
So they see this is exactly why one vaccine for life would be great, because there are many, many people like you, and I am the same, who don't get their vaccine every year. And it just encourages spread of the virus. So in this study, what they did basically was to identify an epitope. So the hemagglutinin glycoprotein of the virus is the main target of neutralizing antibodies. They identified a new epitope in a portion of this protein, glycoprotein, the HA, that's very close to the viral membrane and normally is not very well recognized by B cells. And so when you immunize people with influenza virus, most of the antibodies are directed against the globular head, the membrane distal head of the HA. And this is the most variable portion of the HA protein. Now, this group identified an epitope in the stem, which is highly conserved and also doesn't figure very greatly in the immune response. And these antibodies neutralized many subtypes. So they actually made the antibodies against the H5 subtype, the avian H5 that everybody's worried about. But it also they block infectivity of H5 influenza viruses, but also viruses of 9 other HA subtypes. So that we should say there are 16 known HA subtypes, and there are 2 groups. This is all sequence. I'm sure you have seen these sequence alignments, Chris, but based on the sequence, you can put them into 2 groups. And all of the HAs neutralized by these antibodies fell into one of the 2 groups, and that contains the H5, the H1 hemagglutinin, and the H2 hemagglutinin. So this is very exciting, and it turned out that there was a group also in December that published a very similar piece of work in PLOS, I believe, showing again broadly neutralizing anti-influenza antibodies. So this is quite exciting.
That's right. I thought it was pretty cool. In fact, I know a couple of the guys on the PLOS paper.
Yeah, had you known this was coming down the pipe?
No, I hadn't, and it looks really promising.
I thought both of these papers were incredibly impressive, not only in the result, but the amount of data in each one. The paper that I noticed is Nature Structural and Molecular Biology. They have the description of these antibodies, their neutralization capabilities. They also did a mouse protection experiment and showed that these antibodies are very good at protecting mice, even when you give them after infection. And then they solved the structure of HA complexed with the antibody.
No mean feat.
I mean, that by itself would be a paper normally, but it's all together. So this is a multi-center piece of work here. And this structure is very nice because it shows the antibodies binding to the stem of the hemagglutinin. Now, you may ask why we never saw these antibodies before. This group actually used soluble hemagglutinin to raise the monoclonals, they selected the monoclonals from an expression library, a phage display library. They used soluble hemagglutinin, so the theory is that you get this membrane proximal epitope away from the viral membrane in a soluble form and is better recognized that way.
One of the groups, didn't they select from human memory B cells?
Yeah, the PLOS group selected from human IgM-positive memory B cells, right.
So there must be at least a little bit of recognition. Maybe the immune response to the other parts of the protein just overwhelm it. And so it sort of gets left in the dust and you just don't get enough to protect anything.
You're right. And if you use the right antigen to pull out those rare cDNAs in this case, then you see it. But if you just use virus, which I presume is what everyone else has done, then you can't see it.
Right, and it makes me wonder if normal antibodies, because they were using single chain antibodies,
that's right,
would actually be able to get into that site So even if we could make a new response to the site, if the antibodies would get in there in a normal infection.
A lot of promise here. I think this is a very exciting. Two different groups, really exciting. And it's actually a good demonstration of how collaboration can work really well for these diseases that are hard to crack. You get a lot of good people together. No one does it on their own.
That's right. Yeah, these are big projects, and you need to bring in people with different expertises.
Okay, before we go on, short word from the American Society for Microbiology.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
Now, two stories that I picked in honor of Chris. They are poxvirus stories, because Chris likes poxviruses. He's worked on them, right?
That's right. Yeah, many years.
And the first one I noticed just came out, and it's actually a topic that one of our listeners suggested we cover, and it's using poxvirus as an oncolytic agent. That is, vaccinia virus, which is the lab strain, as an agent for, in this case, killing pancreatic tumors. Had you seen this before?
No, I hadn't, but it's funny again that I know one of the authors, Nanhai Chen, was a postdoc with Mark Fuller at St. Louis, who collaborated with for many years.
So the strain they use here, GLV1H68, can you tell us something about that?
No, I did see a reference to another paper where they talked about the background of it, but I'm guessing it's just a regular vaccinia. It may be one of those with a few deletions more than the others, so it's a little bit less virulent.
Vaccinia is the strain that most people working on pox viruses or human pox viruses will work on. Is that correct?
Well, vaccinia is, I guess, is that a species? Yeah, it must be. But there's a whole variety of different vaccinias. People have developed the original vaccine, Dryvax, was actually a real mixture of viruses. It was never purified. So there's all sorts of different vaccinia-like viruses. I mean, they just had different mutations and deletions and all sorts of things in there. And now people work with specific strains. WR is one of the most common. Copenhagen is another you might hear about.
What is the origin of vaccinia? Do we know?
Not really. There's been debate whether it's horsepox or cowpox, but it was sort of lost in history.
So in this paper, they take this virus. I think their virus has some luciferase or other reporter genes put in so they can monitor them easily in mice.
That's right.
They show that this virus can kill pancreatic cells that are in culture, which is very nice. But then they also have a mouse model. It's a nude mice. They don't have a thymus, so they don't have an immune response. And they put pancreatic tumor cells in the mice. They grow a tumor. And then they inject the virus intravenously, and it kills the tumor. But the interesting thing to me is that it's specific for the tumor cells, and it doesn't seem to replicate anywhere else in the mice. Now, my question for you, Chris, is is vaccinia that restricted in host range normally, that it wouldn't grow in a mouse?
No, you can grow vaccinia in pretty much anything. That was one of the kind of questions I had about this whole thing. They talk about it being targeted, and I'm not sure it is targeted in the way that a lot of people think of, targeting anything, either an antibody to a protein. There's a very specific thing there.
Yeah.
I think what they're more talking about is that it only grows in certain tissues in the mouse.
Exactly, yeah.
I was looking at one of the other papers, and Grant McLennan's got it a bit better when they talk about tropism in the animal. I don't think it really specifically goes to a tissue. It's that tissue can support its replication.
Right. I think, that's absolutely right. It's not a mechanism. It's not a matter of targeting. It's just that the tropism is restricted to that tissue. I'm looking at the table 1 here where they have virus titers and tissue samples, and it's pretty low everywhere except for the tumor. Brain is 0, a little bit in lungs, and a little bit in spleen across all the mice, but the other tissues are pretty low.
Right. I was going to say, I think the idea behind all this is that the tumor tissue is actively growing, and that supports the virus replication better.
Right.
Or there's been some really nice work done with myxoma virus that is much more restricted in host range. Normally it's only rabbits, but in certain human tumors that express or have higher levels of certain key proteins in different pathways, then that allows the virus to grow in a specific cancer, essentially.
Yeah, we should point out that there are many, many viruses that are being considered for this kind of use, oncolysis, and many of them do similar things. They just replicate in the tumor. And I think in general, we really don't understand the basis for that, and in other cases also. And I would think that before you would approve any of these for use in humans, you would understand the basis for that preferential replication, don't you think?
Yeah, I think that would be important to get that basic biology behind it.
Yeah, because if you don't know the mechanism, who knows what kinds of mutations are going to arise that may get around, whatever the restriction is. So you really should know what it is.
Yes, I think the use of vaccinia is the idea that it was used for smallpox, and therefore everybody's kind of happy with that name.
Yeah, well, certainly using a virus that's been in many, many people, you would think they would use Dryvax in the end, right?
Well, Dryvax isn't a good vaccine in that there are various complications. I guess after 9/11, when there was this initiative to vaccinate medical staff around the US, a lot of them started refusing the vaccine because they thought the risk was so low that they didn't want to take complications of Dryvax.
Mm.
And there are now better vaccines with other genes deleted that are safer.
So it's conceivable that one of those derivatives would be used instead of this vaccinia virus, right?
That's right. Or it may be one of those. I didn't have a chance to look at what GLV was.
So there's more work to do here, obviously, but it's just an example of using a virus to kill tumors. We'll go over some others in future TWiVs. But since Chris loves those pox viruses, we thought we would have one using pox.
That's right. One of the early ones, I think, was using reovirus. Patrick Lee in Calgary was the pioneer there.
That's right. Not far from you, right?
That's right. Just down the road when I was in Edmonton.
Yeah, I used to see a lot of those papers. It seems to have quieted down lately, but I know we got an email to TWiV a few weeks ago from a gentleman who asked us to talk about this reovirus use. Apparently it's going forward in a company, so maybe we'll cover that in a future episode.
Right, that may be why we don't hear so much about it under patent wraps.
Yep, it's proprietary now, no more open stuff, right? All right, our last story is another pox virus. It is a paper in Journal of Virology, just came out March 2009, genome comparison of a non-pathogenic myxoma virus field strain with its ancestor, the virulent Lausanne strain. And this all has to do with the use of a myxoma virus, which is a rabbit pox virus, to control rabbits in Australia. Do you know this story, Chris?
Yeah, this is a classic.
Why don't you tell us that? Because I think it's fabulous.
How can I put this? I think it was the 50s. I think it was that early.
1859, yeah.
The use of myxoma was that it grows relatively innocuously in certain rabbits in South America, and I think actually even in North America, some of the myxomas. And it produces kind of a mild disease, small lesions, I think, on that rabbit, and the rabbit doesn't really worry too much about it. But you put the same virus into a European rabbit, and the rabbit is dead within probably about 10 days, with mortality rates close to 100%. And of course, it was the European rabbits that were introduced into Australia and causing terrible destruction of the various farmlands and grasslands. And then, so the idea was to release this myxoma virus, which would become a sort of a natural control agent. It worked quite well, at least for the farmers, not the rabbits, in the early days. And then what happened was that resistant rabbits arose, essentially because the other rabbits were killed, so you're left with a population who were naturally a little resistant. But the curious thing was that you also got attenuated virus coming out. And I think that was kind of a surprise and sort of one of those things that you tend to think that viruses evolve to become more virulent. But actually, in this case, the selection, which is always the real case, is that it's selection for survival. And so the virus that persists a little bit longer in the rabbits, doesn't kill them so fast, actually spreads better. So that was a curious finding out of this whole thing.
Yeah, I understand that the rabbits were introduced originally for hunting purposes in Australia. They took them from Europe and brought them there, right?
That's right.
And they outgrew, and they had to get rid of
rabbits and bullets.
That's so funny. And so then they put this virus in. So this paper, it's the first time I've seen this. I always wondered what was the nature of the changes that this virus underwent in the field. And so this paper looks at that, correct? It compares the virus they introduced with a field strain that they pulled out a few years after introduction, correct?
Yes, yeah. It's a sequence of an attenuated variant, I think from Spain. The authors are from Spain. I think the virus was local as well. Something that I've actually been using to vaccinate rabbits, which is kind of a turnaround from the Australian story.
So actually it's a bit different. It's a strain isolated in Spain in 1995, a naturally attenuated strain, and is a descendant of the virulent one. But presumably the behavior in the rabbit population is a sort of a mimic of what went on in Australia.
Yes, you think so.
Okay, so this is not actually an Australian virus. My mistake, okay.
Well, the same virus was released in Europe.
I see.
So, yeah, in the UK when I was a kid, occasionally you'd come across a rabbit that had myxoma virus infections. And, they're not very pretty. It's kind of not a nice way to die for the rabbits. They get essentially nasal congestion. And I think rabbits are obligate nose breathers, so they sort of suffocate eventually. They get bacterial infections, not so much just the virus, but they get these bacterial infections, and that's what sort of kills them in the end.
So what do we learn from the sequence comparison here?
That there's not that many changes, but it's kind of interesting when you look at it that there are at least 4 gene changes. So they're saying that the attenuated variant is altered in at least 4 genes, that those 4 genes are inactive. So that's 4 genes out of maybe close to 200. And then there'll be a bunch of other minor changes either to protein coding sequences or to control regions.
Mm-hmm. So these genes have just numbers that mean nothing to me, M9L, M36.
That's right.
So I presume they have no known function, right? These are just open reading frames that are translated, but we don't know what they do.
Well, some of them we do. We know a little bit about them. And the key one is one that's, I think it's the M135, which is related to protein that kind of looks like the alpha-beta interferon receptor. So the other ones, they don't see a great tie-in to virulence. A little bit's known about some of those other genes. But this one has been known to be a virulence factor in myxoma and also in vaccinia, because a lot of these viruses share the same set of virulence genes.
That's interesting. So they say the mutation in this gene, M135R, causes it to frame shift, and it breaks the protein into 2 pieces, basically.
Right.
So that would antagonize, presumably, the function, which is probably, as you said, to neutralize interferon, right?
Yes, I think so.
That would do it, I would guess, yeah. So the attenuated virus can't antagonize the interferon system, and it's neutralized by the innate response. So I guess you'd have to put that mutation into the parent virulent virus to really test the hypothesis.
Right, and there could be multiple changes.
Sure.
It's kind of curious that these viruses do have such a lot of virulence genes in them. And when we were working on myxoma, and a lot of the more recent stuff that Grant McFadden has done, is that when you knock almost any one of these genes out, it has an effect on virulence, and the virus becomes attenuated.
Yeah, I've noticed in my writing that the pox viruses in general, as you say, have many, many genes that antagonize immune responses at many levels. Quite amazing. Right,
and different viruses will use those different virulence genes in slightly different ways, in that vaccinia and myxoma, for example, may have some of these genes in common. One of them is the interferon gamma binding protein, worked on that years ago. And in myxoma, The virus makes huge amounts of it and secretes it from infected cells, way more than any of the other pox viruses. But if you go to vaccinia or ectromeliovirus, a mouse pox, those viruses make huge amounts of a chemokine binding protein. So it seems that they have the same set of genes, but they've evolved in different hosts, and those proteins, if they're made at high levels, have an advantage for different viruses. So very small changes in promoter regions can up and down regulate gene expression and sort of tweak the system for different hosts.
Well, that's what you can do when you have a lot of genes. This virus has 159 predicted proteins. That's a lot.
That's a lot compared to polio.
Yeah, polio has 12, right? So it doesn't have a lot of flexibility. Now, I notice here at the end, they say the sequences are available through 2 curated poxvirus sites, poxvirus.org and biovirus.org. But I don't see your site.
We're part of all that.
You are?
Yeah.
I see. I click on biovirus and I go to VBRC, which is your center, right?
Right. Years ago, we started off this whole process, sort of viral bioinformatics, and this is with Elliot Lefkowitz in Alabama, Birmingham campus. And one of the things that we did was develop a bioinformatics sort of resource for pox viruses. And that's where the poxvirus.org comes from. And it's sort of still maintained, but it's now part of biovirus.org, which is a bioinformatics resource for 7 families of viruses. And then I have sort of a little separate piece as well, which is virology.ca. So we're kind of working on this together, and biovirus.org is the sort of umbrella site, which is VBRC, Viral Bioinformatics Resource Center.
So you and Dr. Lefkowitz are the head of these centers, essentially? Is that how it works?
Yep, that's right.
So you wrote a grant years ago to get these started.
That's right, on the pox viruses, and that was actually Mark Buller was a collaborator on that as well. And there was some sequencing involved, and that was funded by NIH, I guess almost 10 years ago. It ran for 5 years, and then we've been doing this other site for more viruses for about the last 5 years, and we're just sort of applied for renewal. So we'll see what happens.
So now you have more viral families. So basically, you curate the sequences for members of these various viral families. Is that correct?
Yes, that's right.
And you're hoping to expand it, I understand.
In the proposal for the renewal, we're supposed to expand that to herpes and really anything that NIH says is important. We also built a dengue site. That was part of our mandate anyway, but with sort of a heightened awareness of dengue and more research, they wanted to have a focus site for dengue. So there's it's denguedb.org site as well.
So how did you get involved in doing this? You're a virologist, right? Did you have computer background at all?
No, I still don't. This was years ago in Grant's lab, and we just started sequencing. And so we were sequencing the ends of Shope fibromavirus, which is a relative of myxoma. And our goal then was to look for a gene that was involved in tumorigenesis with Shope. The idea is that the virus makes these sort of small, sometimes big lesions. And was there a tumor gene in there? And so we started sequencing there. And as you accumulate data, you realize that you need tools to organize it. And it sort of took off from there. Realizing the power of bioinformatics, I guess. We made some interesting findings by doing database searches in those early days. And that kind of led us to realize that, this stuff is important. You can sort of get a good lead on things by careful bioinformatics analysis.
What does it mean to have a curated database?
Right. Because everything is in GenBank, and some things are also organized in NCBI. What we try to do is to curate the genomes with extra information. We will correct sequences that have errors in. We will update the sequences as somebody finds a new gene. You might have noticed that one of those genes in myxoma, or I think it was the vaccinia that was deleted, there was a something 0.5 gene. And that was because somebody finally figured out there was a small open reading frame between two other reading frames. And so they put in an extra gene. And so what we do is go back to all the other viruses and look for that gene and keep things updated. So that's part of it. The other part is building, Elliot's project really, is building a knowledge database in which we scan the literature and try and build a resource, a bit like a mini review, to keep sort of useful information, maybe protein-protein interactions, who's got antibodies to particular proteins, who's made knockouts. So it's a resource for people working on pox viruses or developing vaccines or drugs who may not have a total handle on a particular protein.
So anyone can access these data, correct?
Yep, it's all public.
And they would go where to start exploring?
It's kind of interesting that we do it in two ways. Elliot builds an interface that you would go to for biovirus.org. All his tools are pretty much web interface design. So it's just really browsing web pages. And you go there, you pick the virus group that you want to work on, which virus family, and then it will lead you into menus for picking the viruses or what information is there. But from there, or directly to virology.ca, you can come to my section, and that is a set of tools that are built in Java. They go into a little bit more detail and provide analysis functions. We use the same database, so behind everything there's a single database, but we just go into it in different ways for really supporting different users. Sort of the, I guess, first glance at genomes and just pulling sequences out versus more detailed or hardcore analysis. My group puts a lot of the genomes in and helps with the annotation, and Elliot comes in and does the knowledge database part of that. But it's a single database of sequences, essentially.
So I am currently at what looks like your homepage, Viral Bioinformatics Resource Center at athena.bioc.uvic.ca. So that would be a starting point, right?
Yes.
And I notice that down the page it has Analytical Workbench, which is at the University of Victoria, and then on the right it has the Analysis Web, which is UAB, as you said, the web-based access.
That's right.
And now I notice you don't have influenza virus sequence. Is that done somewhere else?
Yes, that's, I guess, partly done at NCBI, but there's another BRC. Originally, there were 8 bioinformatic resource centers set up. Ours got one set of viruses. Flu went to a different site that also does some other organisms. Then there's another site that does E. coli, another one that does bacterial, things like that.
So an effort is made not to have overlap, correct?
Right, yeah.
You're going to expand, as you said, with this new renewal. And how many families do you hope to have?
Well, we will, in effect, be covering all viruses, or the winner of this contract will be doing all viruses, probably except flu, because flu is such a beast unto itself. There's just so much data and specialized needs of the flu community. So that'll be probably a separate center all the time.
So let's take a hypothetical situation. Someone has sequenced a new isolate. Let's say Ian Lipkin's lab has isolated a new virus, and they want to look in your database and see what it's related to. How would they go about doing that?
Depends on the virus. I guess my first reaction, if it was just a sequence and you want to know what virus it is, it's always best to do something like BLAST, just on the total database. But if you've got a specific virus, and these folks in Spain, when they did the myxoma virus, they contacted us and said, we've just sequenced the genome, we need help in annotation and tools to compare them. We put it in our database right at the very beginning before they really finished it. And they were able to do some of the analysis and comparisons with the tools. So the goal, I think the way I like to phrase it is that the database exists as a way of organizing huge amounts of data. And we have interfaces to get the data out fairly easily And then the tools connect to that database as sort of an interactive pipeline in that I can go to the database and pull out a genome and then a gene and then the gene family. We organize genes into families of orthologs. So I can go to, say, myxoma virus, pull out the DNA polymerase, and then click on another link that will give me all the poxvirus DNA polymerases. Then I can select any ones I like and click on another button, and it creates a multiple alignment, something like that. So things are linked together.
Is this something that virologists could do on their own, or do they usually go to you for help?
They could do it on their own. I think this system offers a lot of advantages in sort of speed. There are things that you can always do by hand, for example, but if you've got a tool that makes it very quick, then you tend to do that experiment essentially. So I can ask questions to the database in a few seconds of what genes are in this poxvirus, but not in another poxvirus. You could do that by hand, but it would take your grad student probably all day to do it, as opposed to a few seconds.
And I presume you're happy to help people if they have trouble.
That's right. Yeah, we try to make things really simple to use. As I said, I've got no programming experience. I want things easy to use myself. So
that's the key, to get it so that you can use it, right?
That's right. I mean, the people in the lab who use it are graduates from microbiology or biochemistry. Mm-hmm. But having programmers sitting right next to them is really useful in that they can really tailor things to how the virologist wants to see stuff, not how a computer programmer wants to present it.
So you have a mix of virologists doing wet experiments and computer people side by side in your lab?
At the moment, there's nobody doing wet bench experiments. I got a collaboration with somebody on flu, and we hope to get back and do some wet lab experiments soon. But at the moment, we're all bioinformatics.
Working on this site, basically.
Yep.
Now, I'm playing on your site here as we talk, and I look at the menu, VBRC tools, organisms, research projects, right at the top. And the VBRC tools is a bit off-putting to me because it has all these acronyms, VOCS, VGO, GIPS, GAT2, GFS.
Right.
So how does one find out what they mean? Just by going to them, or?
Well, if you mouse over them, you'll get a short little
description, yeah. So VGO is Viral Genome Organizer, a Java-based interface for viewing and searching genomes.
That's right. It's sort of our interface to browsing the genome.
I see. So you have to be a little patient because it's a few seconds delay before the key comes up, and I'm not sure everybody would be so patient.
Yeah, we're working on that. That's a little bit of a bug in the system.
You might also want to put a key saying, if you mouse over, you can get more information about what each one is. I don't mean to micromanage you, but I always look, like you, I don't have a lot of computing background, and I always look for simple interfaces. The UI is important, right?
Right. Yep. So if you click on any of those links, it goes to a page that will give you help and what it's used for, and all the pages of help should look very, very similar. The idea is to give this very unified feeling to the site. So
you basically are duplicating the sequence that's in GenBank, right? You bring it all here and you do your own thing with it.
Yes. And we'll sort of update them and annotate them if they're not annotated. So often sequences get put into GenBank with no annotations whatsoever.
Right.
If somebody sequences, say, 50 SARS genomes, they may not bother annotating them, but we will annotate them and put them in this database so it becomes usable information.
So I have just done a new rhinovirus sequence. So you don't do picornaviruses at the moment, but assuming you did, would we contact you and say, hey, we have this new sequence, or we just put it into GenBank and you would pull it out eventually?
Either way, we would pull it out eventually. But if you wanted to get it into the database, we can also create private databases for people who don't want the data released until the publication comes out. Mm-hmm. And we do that quite frequently. We also have programs to help annotate genomes based on related genomes. So, for example, that myxoma genome that came out. So it's kind of a time-consuming effort to go and annotate that whole genome with some of the tools we have, you could probably annotate 95% of it immediately, and it would leave you with just a few genes to check, because it could compare its information to what's known about the current myxoma viruses in the database.
So could you explain the difference now between curating and annotating?
That's a good one. Annotating, I think, is the placing of information on the genome. So the gene is between nucleotide X and Y. I think that would be annotation. Curating, I think that's the way we use that is that it's more of going through the literature and adding other information to that genome. Sort of gene Y has kinase function. I guess that would be more curation. Although some people also call that annotation.
How about saying the sequence here gives rise to virus when placed in cells? In other words, it's an infectious sequence as opposed to many that haven't been tested. Would that be a curation?
Yeah, it could be. I think those words get interchanged a little.
I'm sure other people would like to know the difference, people who aren't using these kinds of tools all the time. Well, I think this is a great resource, and we'll put a link on our TWiV website to this so people can go and explore. I think you're doing an important service for virology.
There's lots of things on there. Some of them are very simple, but again, are time savers. We have a nice little tool for doing dot plots, which are very simple displays of similarity, but they can show things that you just never find by trying to do multiple alignments, for example. And we're more than happy to help people out.
Well, I'm glad you could take the time to explain that to us. That was really useful. I'm going to finish up here with a few emails and our Pics of the week.
Okay.
I have 2 emails this week. One from Scott, who says, "Hi, I came across your great podcast this week in virology while searching for new podcasts in iTunes, and I wanted to tell you that I think it's great. Very informative and very interesting. I have an undergrad degree in computer engineering, but I'm interested in learning more about biology. Virology specifically seems very interesting. I was wondering if you could recommend a good introductory text on microbiology, or if you have any general recommendations for someone interested in learning more about biology. Thanks, and I look forward to the podcasts." So I wrote back to Scott saying, microbiology texts these days are tough because the field is really fragmented so much, virology, immunology, fungi, bacteria, that it's hard to get it all in one textbook, which is usually what they do for medical schools, but it doesn't work so well. So instead of recommending one of those, I would suggest you read a book like Animalcules by Bernard Dixon, The Activities, Impacts, and Investigators of Microbes, which is sort of a historical overview of microbiology.
Okay.
Bernard Dixon's a good writer. He wrote a micro textbook, which was used here for many years for medical students, co-authored by Harold Ginsberg, who was the chair of this department many years ago. I would suggest that, and then if you like it, you can proceed. Now, for virology, of course, nothing beats Principles of Virology, which I wrote, but I know it's a very expensive book.
Maybe a bit hard for a computer scientist.
Yeah, so there is actually, you can download sample chapters, and I'll post a link on the website for that from ASM. But a less expensive book would be Alan Kahn's Principles of Molecular Virology. It's less comprehensive, but much cheaper, and it'll give you a good overview of virology. And then if you think you like it, spring for Principles, or enter the link contest here at TWiV, which you can find details about at our website. Basically, if you post a link to TWiV from your website or blog, you might be able to win a free copy of Principles of Virology, and that contest ends at the end of March. Okay, thanks for that, Scott. And the next one is from Xijing Wei, who says, very simply, please talk about enterovirus in the future. Thank you. So that's great. I would love to. Of course, I love enteroviruses. I work on them, and we will certainly be talking about them more. And now for our science picks of the week. So here at TWiV, we always try and pick a blog, a podcast, and a science book of the week as a way of promoting other good online science information. And our blog of the week is something I found at VRC, the VRC blog by Chris Upton. That would be you, right?
That's right. It's the VBRC.
VBRC blog.
Yeah. You're going to put the bioinformatics in there.
And it's Chris's blog. He puts up news about viruses and various other things, and I like it. So you should check it out.
Yep, I usually browse through a whole pile of RSS feeds and fire them off to people in the lab, and they'll just write a little pointer to it.
So you can go, we'll put a link for that in the show notes, and you can check that out and get the RSS for that, which is what I do. I put all these RSSs into a reader, breeze through them. Science Podcast of the Week is Nova Science Now from WGBH, which is a radio station, of course, in Boston. It's a PBS podcast, public broadcasting system. And, of course, they have many, many podcasts. I think this is the best one on science. It basically irreverent stories, intriguing personalities from the world of science. It's a very well-produced podcast, as you might imagine, from PBS. I really like to pick science podcasts made by individuals like myself, but there aren't that many of them. Since we do one a week, we go through them quickly, and I want them to be good. There are certainly many medical podcasts, so you can read about surgery or spinal cord injuries or nursing, and those are all great, but it's not really science as I define it. If you could help me out, send me your favorite science podcasts, TWiV@twiv.tv. That would be great. And finally, our science book of the week. Maybe you've read this one, Chris. The Hot Zone by Richard Preston.
A long time ago, I think.
Yeah, me too, ages ago. That's, of course, the story of the Ebola Reston outbreak in the lab in Virginia. Monkeys came from the Philippines, and they brought Ebola Reston, and the monkeys died, and some people got infected, although they didn't get sick. Descriptions of historical epidemics of rare and lethal viruses. More hair-raising than anything Hollywood could think of, because it's all true. That's a quote from the book Flap. So it's a really good book. It's older now, but he's a good writer, and the story is fascinating because it's true. It happened. So check that out. And we have, of course, lots of show notes on our website, twiv.tv. We have links to all the stories and links to the books as well at the TWiV bookstore on Amazon. And don't forget, you can find other podcasts on science at two different sites that TWiV is a member of, sciencepodcasters.org and promednetwork.com. So check those out. A few more things just in closing. We put virology news links at Delicious now, delicious.com/TWiV podcast. You can put links there. It's a public feed, and we'll discuss them on TWiV. Or you could post a story on Twitter, put the hash mark or number sign TWiV at the end, and that allows us to search for them, and we can find your stories there. Don't forget the link contest, which I've mentioned. Link to our website, twiv.tv, and win a copy of Principles of Virology, details on twiv.tv. We will be live at the ASM General Meeting in Philadelphia, which is in May 19th, specifically 2 p.m. ASM.org has meeting details. Come join us. We'll do a live TWiV. It's going to be video streamed and recorded, and we'll have a live audience, of course. It should be a lot of fun. And we'll tell you more as the date approaches. Chris, what website should people go to find more about what you're doing?
Probably the biovirus.org.
Biovirus.org.
Yep, or virology.ca. And that brings sort of to my side of things. There's another way of getting it.
All right, so we will put those on the website. You can go find out about Viral Bioinformatics and Chris Upton's contribution to that. Send us your questions and comments, twiv@twiv.tv. Chris, thanks for joining us.
All right, that was really fun. Good to talk to you.
I appreciate the time. You've been listening to This Week in Virology, the podcast about all kinds of viruses except the viruses that infect Windows. We don't talk about those. We talk about the viruses that make you sick, or as someone pointed out, not all the viruses make you sick. The bacteriophages we talked about last time don't make you sick. And of course, we talk about the biological viruses. We'll be back next week. Another TWiV is viral.